This video lecture has been prepared as an assignment during the induction cum orientation program arranged by the Ramanujam College, Delhi University. In this video lecture, I am giving information regarding plant tissue culture. Viewers, let us see what is plant tissue culture. Plant tissue culture is a technique of growing plant cells, tissues, organs, seeds, or other plant parts in a sterile environment on a nutrient medium. Historically, uh, the plant tissue culture idea was first of all came into existence during 1902 when Heberlein put forward the idea of totipotency of plant cells. In 1937, White first time established successful root culture of tomato plant. In 1941, Van Overbeek used coconut milk for growth and development of young Datura embryos. In 1957, Skoog and Miller demonstrated the role of auxin and cytokinin on root and shoot formation in tobacco pith culture. In 1962, Murashik and Skoog introduced the medium for tobacco culture. In 1987, isolation of Bt gene from bacterium Bacillus thuringiensis paved the way for production of transgenic plants. The fundamental principles of plant tissue culture depends upon totipotency and plasticity. Totipotency is the ability of plant cells to regenerate into a whole plant, and plasticity is the ability of plants to alter their metabolism, growth, and development to best suit their environment. What is explant? In plant tissue culture, explants are generally used to initiate the growth of plants in the process of micropropagation. The multicellular tissue fragments are called explants, which are obtained from living plant parts. Explant may originate from wide range of plant tissues such as leaf, stem, root, petiole, cotyledon, embryo, or meristem. Selection of explant. The explant is selected, either it may be haploid or diploid, depending upon the source from where it has been isolated. The plant growth can be achieved in two ways: shoot directly by appropriate medium and by somatic embryogenesis. Culture media. Explants are then usually placed on the surface of solid culture medium, but they are sometimes placed directly on liquid medium when cell suspension cultures are done. Culture media are generally composed of inorganic salts plus few organic nutrients, vitamins, and plant hormones. Steps in plant tissue culture technique. First of all, selection of plants are done. Then, explants are isolated from the selected plants. Next step is sterilization of explants to get it free from any infection from undesired microorganisms. Inoculation of sterilized explants on the nutrient medium. How many types of plant tissue cultures are there? For this, This slide represents that plant tissue culture can be done with the help of meristem, protoplast culture, organ culture, pollen culture, callus culture, cell culture, embryo culture, or, and lastly, seed culture. What is the application of plant tissue culture? The commercial production of plants used as potting, landscape, and florist subjects, to conserve rare or endangered plant species, to screen cells rather than plants for advantageous characteristics like herbicide resistance, stress tolerance, and fourth one is large scale growth of plant cells in liquid culture in bioreactor for proliferation or production of valuable compounds like plant derived secondary metabolites and recombinant proteins such as biopharmaceuticals. Plant tissue culture application to cross distantly related species by protoplast fusion and regeneration of noble hybrids, to produce clean plant material from stock infected by viruses or other pathogens, and production of identical sterile hybrid species can be also obtained. Thank you very much.